Good morning, everyone. Thanks for joining in this morning for the WIN Distinguished Lecture. We are really fortunate to have Professor Jan van Hest, who is the Professor of Bioorganic Chemistry, as well as he's the Director of Institute for Complex Molecular System at TUE Eindhoven in Netherlands. Professor van Hest obtained his PhD in macroorganic chemistry with Professor Bart Meiser. I happened to meet him a few years back from TUE. Then he worked as a postdoc on protein engineering under the supervision of Professor David Tyrrell from the University of Massachusetts. Later, he then joined DSM, is a fairly big chemical industry, as a researcher, and later became the group head for the development of new material concept. In 2000, he joined as a full professor at Radboud University in Nijmegen. Later in 2016, he holds the chair of bioorganic chemistry at TUE. He has graduated a number of PhDs with a number of scientific publications and patents. Also, he is a joint founder of startup, because we have a lot of startup here. So he has an entrepreneur mind with startups like Encapsun, Future Chemistry, Noviosense, and Noviotech. So we are so fortunate to have you, Jan, here, and the stage is yours.
Yeah, thanks, Sushan. That was a very nice introduction. Also for the great hospitality over the past couple of days. I think we've seen excellent science and great facilities, and also great food. So we really, at least I think we all have enjoyed really a lot staying over here. So what I'd like to do today is tell you a little bit about one topic that we do in our group, and that's namely the development of nanoparticles that are responsive and that can be used for photodynamic therapy. Because I go a little bit more detail what we exactly mean with this, I just want to give you an overview of what we do as a group. So the bioorganic chemistry group is in fact a group that has an expertise in synthesis of well-defined molecular building blocks. And we use different methodologies for this, from peptide synthesis to polymer synthesis, but also protein engineering. And with these building blocks, we create well-defined smart compartments that we use for two lines of research. On top, you see our line of research in the field of nanomedicine, where we have a special interest to see how the shape of a particle affects its interaction with living cells and tissues. We have particles that have a semi-permeable shell, and we can load them with enzymes or other catalysts, and we can use them as nanoreactors, even incorporating them in living cells to use them as artificial organelles. And we can make particles that are responsive. They can change their properties based on the environment they're in, for example, the tumor microenvironment, or they can be activated via, for example, light. Well, besides this more applied line of research, there's also a second line of research that we'd like to explore, and that's more fundamental, and is directed on the creation of artificial cells. And we'd like to make artificial cells, micro-sized compartments that have both kind of lifelike structures based on an architecture. So we want to make kind of use of condensates, coacevates that have a density that's similar to the cytoplasm. We make them, we membranize them so they have a compartment structure over there. And we can also load them, for example, with artificial organelles, but also with artificial cytoskeletons. Well, besides that, we want to create structural mimics of living cells. We also like to incorporate lifelike features. And we have a special interest in the communication between artificial cells, the exchange of molecules and proteins. But also, for example, motility is one of the things that has a keen interest. So today, my talk will be focused on one aspect of what we're doing, and that's mostly connected to the field of responsive particles that are shown over here. And this comes down to, in fact, our interest to develop kind of novel approaches how to treat cancer. And of course, we all know that cancer treatment was a very kind of a versatile approach. There are many different approaches, and the most classical ones are shown over here. Of course, it's surgery, chemotherapy, and radiation therapy. But you also know that these methodologies, although they take care of most of the treatments of cancer, they still have their drawbacks, especially if you think about chemotherapy and radiation therapy. Systemic toxicity is really a big issue. Well, of course, surgery is a very invasive technique that you use. So over the years, people have thought about other alternative approaches to make kind of chemotherapy well, more accessible. And of course, probably the holy grail on this is to use immunotherapy, where you can treat the patient's own immune system to recognize cancer cells and remove them from the body. And that would be perfect because you can not only treat the primary tumor, but you can also deal with metastasis very effectively. Beautiful developments going on, but of course, still there steps have to be. Another very interesting kind of treatment is this so-called photodynamic therapy of PDT. So what PDT does is you bring, in fact, three components together that all by themselves are kind of non-toxic to the patient. You use oxygen as, in fact, the reagent that you want to use. You use a laser light source near infrared most of the time, and you use a so-called photosensitizer. And the photosensitizer, upon irradiation by this near infrared light, can, well, become excited and then transfers the energy to oxygen to make singlet oxygen particles that are very reactive, and they react with directly any kind of molecule in the direct vicinity. So if you can bring your PTT photosensitizer in close proximity of a cancer cell, this cancer cell specifically will be killed and not the neighboring kind of healthy cell that's surrounding. And since you use a laser light source, you can of course really target the area that you want to irradiate. So as a result, PDT has very few side effects, and it's a very tailored kind of way of treating, especially of course, solid tumors. So we also wanted to move into this field, and together with colleagues from Dongla University in Shanghai, we first developed kind of a theranostic approach to PDT to also try to make a system that is more effective and also dealing with some of the, well, drawbacks that PDT knows. So for that, we developed this kind of specific nanogel. This nanogel has as following now building block this glycochitosan that you see over here, which has amine groups over here that makes it also pH sensitive. We modify this glycochitosan with a porphyrin unit depicted over here, and this is in fact our photosensitizer. So if you shine the infrared light on this molecule, it gets excited and then it uses its energy to transfer this oxygen. Because of the amphiphilic character that is created by the introduction of this porphyrin, this spontaneously forms nanogel structure. But not only that, what we also do with this system is we add some other nanoparticles within this nanogel, and these are composed out of manganese dioxide that have a catalase enzyme on the surface to stabilize the particles, but also to create some kind of biological activity. So these particles are incorporated into this nanogel, and so we have now kind of this complex system that we use for treatment. And this is how these particles work. So why do we need the different elements to have a very efficient PDT? Well, the glycochitosan, because of its amine character over here, becomes protonated and therefore has a chance to end up in a higher level of accumulation in the tumor tissue, which is most of the time a little bit acidic. So you get a higher accumulation of your particles. That's one benefit of these systems. Furthermore, the photosensitizer, of course, can take care of the process that you turn oxygen into singlet oxygen to create kind of this killing efficacy. But we also use manganese dioxide and catalase to help to make this process more effective. Because if you look at tumor environment, often tumors have low oxygen levels because they are so active, they use up so much oxygen that the oxygen levels are low, so they are hypoxic. And in fact, if you use catalase as enzyme, catalase turns hydrogen peroxide into oxygen, and thereby it increases the oxygen levels inside such a tumor environment. And that, of course, helps if you need to use oxygen for your PDT process. Secondly, singlet oxygen kind of molecules, but also other radicals, are normally scavenged by the normal defense mechanism of the cell, which is this glutathione. This takes away radicals that are formed within the cell, and therefore the cell has a natural defense mechanism against it. To make the cell more vulnerable to this, we use manganese dioxide, because this reacts with glutathione, therefore it lowers the defense of the cell, and that makes the cell more prone to be responsive to the treatment with singlet oxygen. What is also very nice is that kind of this dissociation of manganese dioxide to make manganese ions available, and you can trace then also these particles with MRI imaging. So it's a theranostic approach. So how does this work? Well, here you can see how we can increase the oxygen levels by looking at a probe that is in green if its system is hypoxic. If you treat cells with PBS, we don't see anything happening. If you treat it with a particle that doesn't have this catalase inside, also the green color remains, but if we have catalase present, we see that we increase the levels of oxygen because the hypoxic characters diminish. We can also nicely follow these particles because of with MRI, because of these manganese features, and we can also see that here in the dashed circle, here where the tumor is located, we see also some accumulation of the particles in the tumor. And if you then irradiate this tumor, you see indeed that if you look at the non-treated sample, so without the laser, and this is with the laser, you see that if we have our theranostic nanoparticle present, you see there's the biggest decrease in size of the tumor that we can see. And in all other cases, of course, with PBS, nothing happens to the tumor. If you just have the photosensitizer there, you see also a small decrease because it will not so be effectively accumulated in the tumor tissue. If you have, for example, here a particle that is not stable, the manganese dioxide not stable by catalase, but by just BSA, also there we see a loss of kind of effect because we have still less oxygen in the system. So you need all these components together to get the most efficient treatment. Although this was a kind of nice experiment, and you can see that we can really combine different aspects to make PDT more effective, there are still quite some challenges with PDT. One important challenge is, of course, to make sure that you get good accumulation of your particles within the tumor environment. As you can see from the MRI, there's quite some spread of the particles in other parts of the body. Of course, because you have control where the light source is, that doesn't have to be a huge issue, but you want to see larger level of accumulation inside tissue. Furthermore, what's really important with all treatments with nanomedicine, that particles tend to stick to the periphery of the solid tumor. And of course, if you really want to treat the entire tumor, you should also be able to really penetrate further into the tissue to also reach those cells inside the tumor, the solid tumor. So deep tumor penetration is often really also a bottleneck for nanomedicine. And finally, if you want to deliver drugs inside cells, it's also really important to have efficient cell uptake. So in fact, to take care of these challenges, there are in fact three stories I'd like to tell you today, where we have different approaches, how we can kind of achieve to tackle some of the challenges that we're facing. I'm first going to talk about the peptide porphyrin system that has some of these issues. I will then go to another class of polypeptides based on elastin-like systems. And finally, I will show you some active particles that can also increase PDT activity. So let's start with these peptide porphyrin systems. So the porphyrin in this case is a photosensitizer, but photosensitizers are quite often very hydrophobic and they're very difficult to administer into the body. So they don't really distribute well. So you have to have always some kind of carrier system formulation to make sure that they can be distributed. Well, a simple way to make this possible is modifying the porphyrin unit over here with two dipeptides on both sides over here. And the peptide then brings in more hydrophilicity into the system, and you create kind of amphiphilic system that then self-assembles into a development part. What makes it also interesting from these peptides is that these peptides, of course, are pH sensitive. And of course, we expect that upon protonation of these peptide porphyrin conjugates, a pH change will also lead to a different kind of assembly strategy of these particles. They will change their assembly way. And what we of course hope to achieve is that by changing the assembly into other structures, we hope to improve the kind of accumulation in the tumor site and then still make the particles active, that if you shine kind of 660 nanometers light on it, they can produce effectively single reduction over there. So this quite simple design, in fact, works very effectively. So here we have our particles at physiological conditions, pH 7, and they normally, well, they form well-defined spheres, kind of solid spheres, peptide porphyrin conjugates. And as you can see over here with DLS, we see that in fact these spheres are pretty stable. If we can follow them over this of 12 hours, but also longer times, we don't see really any change in their morphology. So they're colloidally stable particles, as you can also see over here. If you now lower the pH to 6.5 or so to 5, the only thing is really the difference in speed of the change that happens. Then we see that these particles really change their kind of aggregation state. Rather than these spherical particles that you see over here, they form these kind of nanosheets, as you can see with this cryo-EM picture, over here and over there. And of course, what we hope to achieve is that by changing this structure into a nanosheet, they tend to accumulate more effectively within the tumor site via the EPR. So this is also what we then observed when we did this in vivo study. So we injected our particles in the tail vein, and then we looked at how they distributed. And you see that if you follow these particles, the porphyrin is intrinsically fluorescent. So you can nicely look at particles simply by looking at the porphyrin fluorescence. We see that over time, especially after 3 days, it's very good accumulation in the tumor tissue over here. Now you can also follow that over here. You see that indeed the peak is really around 3 days, but in fact you can even follow these particles for longer periods of time. So in fact they're very good kind of fluorescent images, kind of markers that you can follow these particles for a week or even longer. If you look at the uptake, you see that besides, of course, strong uptake in the tumor, you also see, as expected, probably uptake in the liver, and that's normally the clearance of these things. What's a little bit less nice is to see also a kind of uptake in the lungs, and that's normally what you don't want to see with nanomedicine there. So that's something you should work on. But for the rest, you see there's no uptake either in the heart or even in the kidney. So if you then look at how these particles behave, if we shine light on the tumor, then we see that in fact these particles are very effective in PDT. So here we have in fact 3 controls and 1 with our active particles. So here, 1 control is just the addition of glucose in aqueous solution, so there's no any photosensitizer in there. Here we have just the particles, but without a laser treatment. This is just a laser treatment without particles, and this is why we have both the laser and the particles present. And you can nicely, you can see indeed that in all 3 cases over here, the tumors are, well, remain more or less what they are. But in this case, in fact, we were not able to take out the tumor because it was more or less gone. So it's a very effective treatment. You see also this scarring over here, which is in fact the necrotic tissue of the tumor that starts to disappear. Furthermore, the particles are not toxic to the mice because they keep their body weight. And here you can see again how the volume changes of the tumor. So in our case, these ones are very effective in removing the tumor from the tumor. Still, if you look at the picture of the distribution, you see that it takes 3 days for these particles to end up effectively in the tumor tissue. And in fact, they also end up in certain other tissues like the lungs, and it's something we'd like to prevent, in fact. So therefore, we're to our next generation of peptide ponderings, and we want to investigate if we could make particles that show a more effective accumulation behavior inside tumor tissue. And we changed our design a little bit. Of course, still the porphyrin is in the middle as our kind of hydrophobic unit. But now we use histidines as our kind of dipeptide because they also have a pH effect that is closer to what you find in the endosomes, also in tumor tissue, like 6.5. And we modified these dipeptides now with a polypeptide tail to make them kind of better water soluble and to get them also better control over the assembly features. And if you then see what happens, then we see that at pH 7.4, now we don't get spherical particles, but we get these very small nanoworms that are formed. That's very interesting to see. And if you then go to lower pH, these nanoworms aggregate. And in fact, they form a reversible aggregate. So they really cluster together at lower pH. And you can also see that from the DLS. So these are our nanoworms over here, and these are large aggregates. So DLS values are not that reliable anymore, but you can nicely see also here with cryo that aggregation takes place at lower pH. So what's the consequence of the behavior of these particles when we do in vivo testing? Well, first of all, how do we do cell uptake? Is also, of course, important. In this case, we can nicely see how these particles behave when they are taken up by cells, and also see if there's a pH effect. So here we have 2 cell types that take these particles up, and we look at them at different pH 7.4 and 6.3. And in fact, in both cases, you see good uptake of the particles, which are, of course, again, using the fluorescence of the porphyrin visible with red, kind of red color over here. And if you then look at the difference in pH, you see that there's some kind of increase in uptake, a slight increase, you could say, when the particles go to lower pH. Where do they end up in cells? Well, you can do that by staining either the lysosomes or the endoplasmatic reticulum. And you see that in both cases, there's some overlap. So it shows that particles end up in lysosomes, which you would expect. They're taken up by endocytosis. They also end up partly in the ER. They don't end up in mitochondria or other organs. So it's very direct to these 2 types of organelles that these particles end up. So if you then look at the accumulation, then we see that this kind of structure has a much more effective way of being accumulated in the tissue. So now already you see that, indeed, if you now look at 3 days, and in fact you're already over your top, already after say about 12 hours or 24 hours, you see that the tumor has accumulated, well, the majority of the particles here. And you can also see that now, in fact, the lung looks much cleaner than it looked in the other case. You still now see also, of course, uptake in the spleen. And now we see also some uptake in the kidneys, probably because these are small nanoworms that probably also therefore, because of the size, can more easily be secreted by the kidneys. So if you now use these particles for treatment, then in fact we see more or less similar behavior as with the other. Again, irradiating the tumor shows that in case over here, we really get rid of the tumors. In this case, maybe not so effectively as the other case, but I think that's just really in the level of significance, which you can nicely see the big difference between the controls and the ones where we use our laser light. And you can also see that the growth of the tumor is really strongly diminished and almost completely eradicated. And this shows that these particles, in fact, are very interesting because they have a good circulation time, these worms, but they accumulate effectively in the tumor tissue. They can really help to treat these solid tumors. Furthermore, what's also nice is that the fluorescence that we have of these particles can also be used as a tool to follow the treatment, because you see that over time, after radiation, the first couple of hours, you still see the particles present, but at a certain moment when the tumor tissue disappears, also the particles will disappear. And the loss of fluorescence is an indication that your tumor is really shrinking and disappearing from the mouse. So therefore, it's a nice theranostic approach again to use these peptide fulcrum particles, not only for good accumulation in the tumor, but you can also use them for effective imaging of how the treatment works. Okay, second topic I'd like to discuss with you is a system in which we go to a slightly larger type of structure. So here, the first I've been talking about small peptides communicated with porphyrins. Now I'm going to talk about peptides that are larger, maybe polypeptides that are based on elastin. So what are elastin-like polypeptides? Well, elastin-like polypeptides are in fact kind of originating from the structural protein elastin that is part of the protein, the ECM of our body, taking care of elasticity of our tissue, for example, your skin or your cardiovascular. It's composed out of different domains, and one of these domains in elastin is called the hydrophobic domain, and it's recognized by or characterized by a repeat of 5 amino acids: valine, proline, glycine, the 4th amino acid, another glycine. And the interesting thing about the system is if you make a polymer out of this pentapeptide, then this polymer has lower critical solution temperature behavior, meaning that at low temperatures in water, it's a random coil and fully water soluble. But if you increase the temperature at a certain moment, it goes through its transition temperature and it becomes a beta spiral with exposure of the hydrophobic side chains. And that then leads to the great aggregation of these polymers. So it's like polynipam, but then the biosynthetic variant of this. What is maybe nicer than polynipam is that we can really control the temperature at which the transition occurs. Because by changing this 4th amino acid, by protein engineering for a more hydrophilic amino acid, you can push the transition temperature up. If you put in a more hydrophobic one, the temperature goes down. You can also further control the transition temperature by controlling the length of the polypeptide. So if you make longer polypeptides, the temperature will also go down. And indeed, it's also concentration dependent. So we have a couple of features with which we can play to control the transition temperature of this elastin-like polypeptide. What's also really nice about this system is that this aggregation is fully reversible. So if you have a cold solution of your polymer, it's water soluble and therefore it's just a transparent aqueous solution. If you go up in temperature, you get, of course, aggregation and precipitation. But if you cool down again, you go back into solution. And this is totally reversible for many times. What you can also do is you can make a very nice trick with protein engineering that you combine 2 different elastin-like polypeptides in one and the same polypeptide. And then you get this block dipolypeptide. And one of these blocks, in this case the blue one, has the amino acids on the 4th position has amino acids that are more hydrophobic, while the green one has amino acids on the 4th position that are more hydrophilic. So if you now start to heat up the polymer in solution, then first the blue block will go through its transition temperature, while the green one will remain soluble. So if you're just above the transition temperature of the blue block, you make well-defined micelles or plasmate-like structures with a colloidal state. If you go above the transition temperature of the green block, then you get full aggregation. And cooling down, you go back from an aggregated state to micelles and back to individually dissolved polymers. So this allows you to assemble nanoparticles in water just simply by controlling temperature or by controlling, for example, also salt concentration. That's also a possibility you can do. What's also nice, because we make these polymers by protein engineering, is that we have, of course, full control of the amino acid sequence. And we can not only incorporate these structural proteins, but we can also functionalize them. For example, we can connect a nanobody, a single chain antibody, to one of the chain ends, namely the one that sticks out. And therefore, we can incorporate targeting functions very easily, simply by protein engineering. And since we have full control of the chemical composition, because we know exactly which amino acids are in the sequence, we can also, for example, know where the reactive groups are. So we can make a polypeptide that's maybe 50 kilodaltons in size, but that only has one amine group, namely at the end. We can use that for the functionalization, for example, with a chemical handle in this case, photosensitizers. So you can make a particle that is suitable for PDT, that has targeting capability, and also brings the photosensitizer very effectively with itself. And of course, then you should be able to use these particles that kill cells that have receptors for the nanobody, while the cell next to it is not affected by the treatment. What's also very nice about ELPs is that they, first of all, can be expressed very effectively via bacterial expression, but the purification is also very easy because you can use this transition temperature. Because if you have elastine-like polypeptides in this protein lysate that you get when you produce them in bacteria, then just by simple adding salt to the system or heating up, your protein will precipitate while the other bacterial proteins will remain in solution and you can take them out. Of course, there will be also some bacterial proteins precipitated, but if you cool down, the ELP goes back in solution and the other precipitated proteins will stay behind. And so by having this cycle a couple of times, you very easily can purify elastine-like polypeptide without having to go through complicated chromatography techniques. So it's a very effective polymer of protein, now polypeptides, to work with. And in the end, you really can see that you make a very well-defined building block. Here you can see an SAS page, so where you see just one band. They colonize so effectively because they often don't have many functional groups in side chains, so therefore you have to often use silver staining to make them visible. But here, this is a band that is pretty strong concentration of the ELP. But of course, if you look at mass spec, you can really nicely see just one molecular weight polymer. And of course, from a polymer chemistry point of view, and I'm a polymer chemist, it's always like a dream because you just don't have any polydissociation, it's just one perfectly defined polymer that you. What's also really interesting about the system is that ELPs allow themselves to be co-assembled. So if I take a mixture of two different ELPs, so say a diblock and a monoblock, and which, of course, the monoblock has a certain transition temperature, and the red block of the diblock has a certain transition temperature. And I heat this mixture up above the transition temperature of the less hydrophobic block, so in this case, say, for example, the red one, then they will assemble together in one and the same particle. If I do this slowly, then I can first go, for example, into the assembly of the black block, then I get an aggregate, and then only the micelle. So I can, in fact, assemble particles in different ways based on the process, the kinetic process that I follow. So I can either go to a co-assembly state where I have a mixture of ELPs being part of the same particle, or if I heat slowly, I can have them aggregate separately from each other. And I first go to the position of the black block, and then only to the position of the red block, and then you have separate particles. So this is really interesting because now you can just take two ELPs with different kind of hydrophobic domains. It doesn't really matter how they're composed. The only thing you need to know is that transition temperature. Then you can mix them and then let them assemble into one and the same particle. You can nicely see that also if you look at aggregation of these structures. If you go for a slow heating, then at a certain moment, if you go through the transition temperature of the first block, the black hydrophobic block, you see an increase in kind of turbidity because that block precipitates and that gives turbidity, of course. And then later on, if you go through the transition of the second block, you get the formation of the micelles, which you hardly see them, of course. If you do a very quick heating, then in fact you see that nothing happens. You very quickly heat to say 37 degrees. You kind of circumvent the aggregation of the black block. You just aggregate everything together in micelle, and the black block is in the center of the micelle. And therefore, you make a very stable particle that's over there. So why I'm telling you this? Well, we can use this phenomenon to incorporate two different blocks with different features in one and the same particle, and that can be very useful for further deeper tumor penetration. So what we do is we co-assemble different types of elastine-like polypeptides. We first take this diblock elastine-like polypeptides, which is say the core of our particle that we want to have. We can incorporate an antibody for targeting purposes. So this is in fact the same structure as this one. And then for the functionalization, we introduce this monoblock that is functionalized with the photosensitizer. CT1 is a photosensitized form. What we now can do is that upon rapid heating, and only upon rapid heating, all these three combinations of these three polymers are inserted into the same particle. And then they form a structure like this. It's a targeted nanoparticle, of which the one that doesn't have a targeting unit, so we have non-targeted particles. But The photosensitizer is in the core in both cases of our ELP nanoparticle. Well, this is not so special. We could also have done this in different ways, but what we now can do is we can make this ELP special. We can make this ELP namely photoresponsive. And then we can do that by the incorporation of a methionine, one of the natural amino acids that has a styrether over here. And this styrether is in fact also responsive to oxygen, single oxygen species. So if I now shine light on the photosensitizer and I make single oxygen in the core of my cell, then this sulfur, this styrether, becomes oxidized. And what happens then is that this is more hydrophobic, this is more hydrophilic, and we change the polarity of this block. And as a result, we trigger a different type of assembly of the particle. Simply by using the same light that we use for the treatment, we can go from a bigger particle to a smaller particle because we change the features of this monoblock over here, making it more hydrophilic. Now, how does this look like? These are the components that we use. These are letter codes. So this is more the alanine glycine containing hydrophilic block. This one contains valine and phenylalanine in the hydrophobic block. And the other block over here that carries the photosensitizer has methionine over here for the oxidation purposes. So this is the CT1 phthalocyanine. That of course is our photosynthesis. So we mix them together by heating up quickly. And then we use photooxidation. And indeed, upon photooxidation, you see that the transition temperature of this block over here is really increased. It goes from, say, 28 to 34 degrees if you go a certain concentration. There's a slight concentration dependence, as we know, but indeed, you can nicely see there's an increase about 6 degrees in temperature upon oxidation. So there's a very different component. Well, how does this affect the assembly process of these particles? Well, here's what you see for the two different particles. So these are the non-targeted nanoparticles, and these are the particles that have a target unit over on the surface. And over here, these are the normal ones, MP1 and MP3 over here are the ones that, in fact, are the original particles, and MP2 and MP4 are the ones that are the result of irradiation for a small time, to oxidize the particles. And we see that the DLS, we go from a larger particle, say around 100 nanometers, to one that is about, say, 20 nanometers. And that happens in both cases, very reliable. Difficult to see them in cryo-EM, but still, we have very good cryo-EM facility, and here we can see the normal micellar-like structures of the normal particles that are not irradiated. And if you look very carefully, and I guess you have to believe me for that maybe, you see the smaller, what we call nanoclusters of the MP2 and MP4 particles that are formed. They're not single molecules, they're still clusters. And why do they form clusters? Well, the reason for this, maybe I should go back to this slide, is the fact that we use over here in our structure this phenylalanine as an aromatic unit. And that's not done for nothing, because this phenylalanine will interact with this aromatic phthalocyanine. So as a result, there's some kind of interaction still left between the dye block over here and this now hydrophilic methionine containing block, because of the pi-pi interaction between T2-1 and the phenylalanine, which then keeps these two ELPs together into not a big structure anymore, but a nanocluster. And it's not only that we see this with cryo-EM, but we can also see that with the functionalization of these systems. Because here we have the two functionalized nanoparticles, and these are the two that don't carry any ligand on the surface. And so if you then look at uptake, we see that these are not taken up effectively inside cell, but these ones are. And in fact, both the normal particle that was composed indeed out of this kind of nanobody containing ELP structure, but also with the TT1 in here, that's colored in red, you see that both end up into the particle, into the cell. But also this one, this nanocluster, shows in fact the nanobody, but it also shows the phthalocyanine, and both again are taken up by cells. And this demonstrates that this nanocluster has both the targeting ability still, and it has the ability to use PDT, to be used for PDT, because it carries the phthalocyanine structure. So we have now a particle that we can infect and see to change in size without losing its functionality. So we have a bigger particle that is very efficient for biodistribution, for circulation, but the moment it reaches the tumor site and we start irradiating the tumor, the particle goes from, say, 100 nanometer size particle to 20 nanometer size particle, and that of course will increase the penetration ability of these particles. And we've demonstrated that using a spheroid, so a kind of a tumor spheroid that's grown out of tumor cells, a 3D kind of image to show that this happens. And you see a couple of effects over here. We see indeed that a very good uptake is only shown if we have the targeting ability of these particles. And you see that, in fact, that's the case over here with these two that are taken up effectively. We also see that size matters. So if you have smaller particles, they have a better way to penetrate the part of the system than the bigger ones. In this case, of course, we made the particles simply by irradiation, then add them to the tumor spheroids. So we didn't do this in situ yet, but you see that this effect shows quite important differences in how they behave. And if you look at the fluorescence over here, if you quantify this further, you see that our single a nanocluster that carries the targeting ligands of the nanobody is in fact the most effective one in bringing the photosensitizer into the tumor tissue. If you then look at treatment, and you just then irradiate for a longer time, take a 5-minute time frame, then we see that our particles in fact are very effective in killing the cells, especially the particles that's depicted over here, the MP3 and MP4 ones. And what's also really interesting is that we can start with the bigger particles, MP3, we add them to cells, we irradiate for a short amount of time, we therefore shrink the particles to a smaller size, we wait for further kind of penetration of these particles, and then we irradiate a second time, keeping the total irradiation time the same. But then we see indeed that we still can gain further in cell killing. We're still a little bit more effective in this case than when we just add the small particles to the cells. So it shows that it has a much better type of taking these particles. Okay, so final topic that I'd like to discuss with you is another way of kind of trying to get particles further into tissue. And one way to do this is to make particles active. Most approaches that I've shown you now, in fact, all approaches are based on passive accumulation, either taking kind of taking use of the APR effect or some kind of pH change, so the particles stick to a more acid environment. But of course, it would be even nicer if you could design particles that have the ability to push themselves further into tumor, into interstitial tissue. And for that, in fact, there's kind of an entire field now open there, and there's a field of nanomotors that has the ability to go further because they haven't built in into the kind of energy supply that allows them to move around. So how can we make these things? Well, I have many different approaches, but one approach that we followed to make these particles I'd like to show you over here. Again, first a little bit of problem synthesis, how we can make our building blocks to create these kinds of nanomotors. In this case, the building block that we make is based on polyethylene glycol as our hydrophilic block. It's of course a often used system because of its kind of biological capability of kind of its stealth properties. And then we use this trimethylene carbonate monomer that is functionalized with this pentafluorophenyl ester group over here. This work is done by Jim Hendrick's group already quite some time ago, and they've shown that if you polarize this group, you make this trimethylene carbonate block, and you have this activated ester in the side chain. And now this activated ester is very effectively modified if you add any kind of primary amine to the system. And we've also used this technology, and it works beautifully. In fact, what we did, you can use simple modifications, but in our case, we decided to incorporate this specific molecule. Here's the amine. This will react with the activated ester, then you make the amide link, and then you introduce this specific molecule. It's called an aggregation-induced emission moiety. That's in fact the kind of work pioneered by Benzon Tang, Hong Kong University, BNU, also for biological applications. And it has some very interesting features. If you look at the structure, it's a highly conjugated aromatic structure, as you would expect. And because of that, you would expect this molecule to be highly fluorescent, but it's not. And the reason it's not has to do with this so-called tetraphenylethyl moiety that's over here. This TPE group is namely very flexible, and rather than being flat in the structure, it easily just starts to distort, and therefore it breaks the kind of the aromaticity of the system, and you lose the conjugation. So if this TPE group is free in solution, it flips around, and therefore you have no or very low fluorescence in the system. But then it's very interesting, if this molecule is losing its flexibility, namely when it becomes aggregated, assembled, then it stacks with other kind of TPE groups, and then suddenly you have this conjugated structure intact, and therefore they become fluorescent. So one of the issues, of course, always with fluorescent molecules is that they quench upon aggregation. This group of molecules has the opposite. They are only fluorescent the moment they are aggregated. And so what's easier for us than to put them into the side chain of a hydrophobic domain of a blockopolymer, and then by assembling into vesicles, we create intrinsically fluorescent systems. And that's also what you see over here. So we can make these kind of diblocopolymers. We let them assemble into polymersomes, and then here you can see these polymersomes with cryo-EM. And if you then look at the fluorescence, then in the MSO, where the blocopolymer is fully disassembled, there's no fluorescence over here. But if you keep on adding water, you see at a certain point, indeed, assembly takes place, and they become highly fluorescent. So it's not only interesting for these systems to be highly fluorescent, what's also really interesting is that these kinds of molecules are also photosensitized. So they can not only use their excited state to give fluorescence, you can also use them to transfer the energy to singular oxygen. So as a result, they can be used for PDT treatment. But I already mentioned, we want to use these systems for nanomotor applications. And for nanomotor applications, it's very important that if you want to move something forward, you should be able to put a force onto a particle that is kind of asymmetric. If you place the force on the particle on all sides equally strongly, isotropically, then the particle will not move. And how can you now make a particle move? Well, either you make a particle that is intrinsically asymmetric, and as a result, therefore, it has a chance to move, or you place the force or the force generating moiety on the particle in an asymmetric way. And one effective way to do this is by making so-called Janus nanoparticles. And that's what we did in this case. So we made spherical polymersomes out of here. In that respect, of course, these are isotropically, of course, on all sides the same, so there's no anisotropy in the system. But if we place them on a substrate and we let a layer of gold be spotted on the surface of these particles, of course, only the exposed side of the polymersomes will get some gold layer, nanoshell, while the bottom side will be shielded from, say, the gold rain, and therefore will be not coated. And here you can see, in fact, that you therefore create these kinds of Janus-like structures. And this is how you can proceed them. So this is the AE polymosome that has this Janus nanogold layer over here. And now, in fact, the third very interesting element comes along from these AIE moieties. So they can be used for intrinsic fluorescence, they can be used for PDT, but now they also have the ability to interact with the gold layer. Because if they get excited with 300, 450 nanometers, they will fluoresce, and that energy effect can be taken up by the gold layer that is then used to turn that energy, that wavelength, into heat. And then you create a photothermal effect. So the gold layer over here leads to the creation of photothermal effect. You can also do this directly by irradiating the particles with a wavelength of 760. That also gold will be activated, but we also have the synergistic effect that also irradiation with this wavelength will lead to transfer of energy to gold, and also therefore leads to motility. So this is in fact the idea that we have with this particle. We have this Janus particle, we shine light on it, we in fact use 2 photons, the infrared light, that leads to the activation of this gold layer, photothermal activation, and hopefully therefore we're able to either penetrate cells more effectively or also penetrate human tissue more deeply. And at the same time, of course, we have the AIE there to produce PDT, thereby kill the cells effectively. Well, this works very well. I mean, here you can see the motility of these particles, so we can track these particles in the y and x direction, the mean square displacement. If you don't have a light source, they just show Brownian motion, so they just move around the place where they are. But here you can clearly see that there's a motility on here, there's enhanced motility of the particles upon irradiation. And if you then look at a biological activity, well, here you can see what we do. We have our particles in presence of cells. It's a 2D cell culture. In blue, you can see the cell nuclei, so you know where the cells are located. And then we have, in fact, 2 stains. One stain for healthy cells, that's green, and the other stain for dead cells, that's red. And in fact, we irradiate this square, and well, you can nicely see that only the cells in the square that are irradiated are really dead. So it shows really effectively that these nanomotors are effective cell-killing agents. However, you can also do this with a regular PTT system. What makes this more interesting is the fact that if you look at the 3D spheroid system, and we look there at, well, cell death in this system, that we see that if we have our gold nanoparticles over there, and we shine light on them, that they have a better ability to penetrate this tissue, the spheroid, and therefore show a higher level of cell killing than in our countries, demonstrating that, in fact, there's better tumor penetration possible. So this is one design, but we can make also other designs with nanomotors. And in fact, this is the last topic I'd just like to discuss with you, where in fact we now don't use a spherical polymerisome, but we use a bowl-shaped polymerisome or sematicide. I also mentioned that yesterday during the workshop. How can you make these systems? Well, in this case, we use just polylactic acid, a block of polymers with polyethylene glycol. We dissolve these block of polymers in organic solvent, dioxin and THF. And then we add water to the system, and then if you add more water, at a certain moment, the polylactic acid becomes insoluble in solution, and then it starts to assemble into well-defined polymeric vesicles. Upon doing so, you get a polymerisome, a polymeric vesicle, that have inside not only water, but also still this organic solvent. And then we can do dialysis. We can remove the organic solvent from the vesicle very effectively, but as a result, because you have about 50% of organic solvent volume in there, the vesicle empties out, because organic solvent leaves much faster than water can get back inside, simply because of the hydrophobic barrier that is created by this polylactic acid membrane. If you lose volume inside, then the extra surface area has to go somewhere. And there are two ways to go. They can either form a prolate structure, so that's a tube-like structure, but they can also form a disc-like structure that then fills up with water again, and that ends up with this kind of bowl-shaped structure, where we have a new cavity in here that is in direct connection with the outside world via this mouth region, and therefore these particles are also known as stomatocytes. It's also shown by, for example, for liposomes, so we didn't invent its name. It's a known name, stomatocytes, from the Greek word of stoma, or stomach, if you like, and it's recognizable also in here, shows the kind of the morphology of the particles. The difference with liposomes, by the way, is that this morphology can be frozen in if you take enough of organic solvent away, because then the membrane becomes stable and the particles can be isolated and used also on the physiological condition. So now we have an asymmetric shape. And we thought, okay, let's use this shape to cover these particles with also photothermal agent, in this case gold, but not a gold layer, what we did with sputtering, but we want to do gold nanoparticles. One of the reasons also is that gold sputtering looks nice with genus nanoparticles, but you can imagine that the scalability is pretty low, because it's difficult to, well, you just have a monolayer of particles on the surface, and well, gives you lots of problems with scalability. So this is easier because you can just do this in solution. You can grow the nanoparticles, the gold nanoparticles, on the surface of these stomatocytes. To be able to do so, we have the immune group over here. And then what we have is a particle that, again, is asymmetric, and therefore we expect these particles also upon the radiation to show motility. And in this case, we were specifically interested to see if this could help in getting these particles more effectively inside the cell. Well, this is how these particles look like. We have kind of analyzed them extensively with cryo-electron microscopy. This is a normal spherical polymerisome, as you can see. This is our stomatocyte, so this is the original volume that was over here, that upon dialysis is kind of diminished, and this is the new cavity with here the opening to the outside world. So this is now our stomatocyte structure. And here you can see the gold nanoparticles on the surface of such a stomatocyte. So you can see very well distributed gold nanoparticles over there. And you can also see that with absorbers that affect gold is on the surface, but I think this probably already tells you that that's the case. And that effect, we were surprised, because we thought this would be just an easier way to make a genus-type particle with gold, and would expect similar behavior of these stomatocytes compared to the genus nanoparticles that we made before. Maybe even less effective, because, well, the spread of the particles on the surface would maybe lead to a less strongly separation between where the gold is and where the gold is not. But then if you look at the photothermal effect of the system, then we saw that this was pretty hard. So we saw over here, upon irradiation with different wavelengths and with different strengths of the laser light intensity, we see that we really can increase the temperature well, up to 25 degrees in solution. And this is a very reproducible process, showing indeed these particles are photothermal. That's one thing to see. But then, to our surprise, these are very effective movements. So here you can see their displacement. Here you can see how these particles move. So here we irradiate, and then these particles, they shoot around like rockets. And this is in fact the difference between the on and the off state of these particles. And if you do the calculations, you see that these particles move around with about 100 microns a second. And that's for a 200-300 nanometer particle very fast. If you do the calculation at microscopic levels, we will probably see that it's very, very fast. What's also nice to see is that it's not so much affected by the medium. You do these experiments, both in ILIQ and in DMM, so growth culture medium, you see that utility is more or less kept. Maybe nicer to really see how this behaves. So here we have in fact these particles. This is nanoparticle tracking analysis, and we irradiate here from this side, and here we irradiate from that side. So we can show this phototactic behavior. We can really push them in a certain direction. And with these kinds of techniques, we can analyze the motility of the particles very effectively. So we have a very fast moving particle, but in fact we were still kind of a little bit flustered by why this is the case. So in order to find out how we could expect or explain this effective behavior, we did a very thorough analysis of how these gold nanoparticles are distributed around our stomachs. So we did this full 3D analysis cryo-EM. We in fact located all the gold nanoparticles, we counted them all, we pictured them all, and then we looked at the distribution of the gold nanoparticles, and we used that as input to calculate, say, the photothermal effect of these gold nanoparticles at different places around the stomatocyte. There you see over here, that's in fact a simulation that we could do, is that you see there's a hot spot over here, at this side, at the bottom of our stomatocyte, where the temperature increase is much higher than in the rest of the stomatocyte. And that, of course, explains now why these particles are such effective movements. Okay. What do we do in a biological concept? Well, also there, they work very effectively. I already mentioned that we were interested here to see how they can be used for effective cell uptake. So what we did is we took our cell types and we added our nanomotors to this. And in solution, in the medium, next to the cells, we added either siRNA or we added a protein, fluorescent related to both cases. And then we started irradiating at the sample for a few seconds. And what we observed is that these particles have the ability to directly penetrate the cell. And by doing so, they create a transient opening in the cell membrane. And that allows also molecules that are in the surrounding solution to leak into the cell and enter the cell and be taken up by the cell. So in fact, what we have now over here is a very efficient method to get things into the cytoplasm via this motility, via this motor system. So you pinch a small hole in the cell membrane, and in fact, fluid gets in. In this case, in fact, the cargo is still outside of the nanomotor, but of course, we also work on methods to place cargo inside the nanomotor. So the nanomotor directly delivers the cargo inside the living cell. Just to show you how this, in fact, also how quickly this works. So this is an example of one of our systems that can be used for direct cell uptake. So BSA is fluorescently labeled, it's outside of the cells, it's not taken up in the cells. So the black spots over here are our cells. And now we start irradiating the system, and then we see that directly BSA gets inside. So it's a very effective delivery method into the cytoplasm using a nanomotor that pinches a hole inside. Of course, you should not exaggerate how much energy you put into this, because otherwise you will fry the cell. So you have to be a little bit careful with dosing the amount of energy. But if you're careful about that, you're able to really get these things inside cells. And of course, we tested that then the cells remain viable after that treatment. Okay, this brings me to the end of my talk. I hope I've shown you that with design and responsive particles, we have the ability to make particles that have a better way of penetrating tissue, being more effective in photodynamic therapy. And of course, this is one of the things we'd like to investigate. Besides interest in kind of cancer therapy, we also like to develop our systems, for example, for immunological purposes. And in the end, we really like to see if you could combine PDT with immunology to see if we can really get combination therapies working that hopefully then are not only active against primary tumors, but also against metastases in an effective way. So I'd like to thank the group who contributed to this special mentioning of a number of people who worked on this, and also a couple of universities, Sunway University, Radboud University Medical Center, University of Bordeaux and Madrid, Guangxi Medical University, and Chinese Academy of Science in Beijing for quite a lot of animal studies you've also seen. And finally, I'd like to thank you for your attention and happy to answer any.
Thank you so much. If no question, let's thank the